course. All right. So little question to kind of recall what we did yesterday. First off, just a couple of things for the week. Uh, Friday is going to be our photosynthesis test. We should be all good by then. And by tomorrow, I think we'll be through most of the notes. We have a lab for photosynthesis we're going to be doing this week. So on our lab day, we'll do that. And then, yeah, just keep an eye out for review stuff that I post. And also, um, make sure your choice board, all that stuff is up to date and you have something to use. And I think that's about it. We'll see. Next week's the end of the marking period, so I'm going to be trying to get grades all updated. The It's also a little bit choppy next week. We're not here Monday, and then there's a delayed opening for in-service for teachers on Tuesday, I think. And then there's an extended homeroom or something Friday. So, yeah, keeping it really consistent for the end of the marking period. So just be aware next week's going to be a little bit weird, and it's a 4-day week. All right, so are there any questions at all? Okay, so we left off talking about kind of phase one of photosynthesis was the light reactions. We're going to get into the light independent reactions today, but we still have a little bit to talk about the light reactions that I want to get into our drawings. Let's answer this question. That'll lead us into a review of what we did, and then we'll add a little bit. So if we have a situation where if we look back at the thylakoid, where the hydrogen ions are leaking out of the thylakoid. So if this was our thylakoid, we have hydrogen ions or protons, same thing in this case, and they're leaking out faster than they're being put in. What's going to be the consequence for the light reactions? Yeah.
Would the reaction use like the H plus ions necessary for the ATP production?
Exactly. So they're not going to be able to produce as much ATP. Why is that? Remember, having that difference of hydrogen ion concentration is important because that's what powers the ATP synthase that sends ATP to the Calvin cycle. So what do we call that difference in concentration of hydrogen? Yeah, that's the proton gradient. So the proton gradient, just like in cellular respiration, is really important. If you have an equal amount, this movement won't happen. It's kind of like gravity. Things naturally move from high to low. And the ATP synthase is taking advantage of that to change and use that energy to make ATP. So if you don't have that gradient, no ATP will be made because the ATP is really created by that first electron transport chain that we highlighted here. That's what keeps things going because the electron transport chain is constantly moving hydrogen into the thylakoid. So even though some is coming out into the stroma, it's always being pumped back into the thylakoid to keep the gradient in place. And that's why plants need water and sunlight to keep that process going. And that's how the ATP gets generated. So the proton gradient and this first electron transport chain, if you didn't have that, you wouldn't have the gradient, then you wouldn't have ATP production. So does that make sense? Questions? All right, let's look at a quick model of where we left off with the light reactions, and then we're going to make a little segment on our drawing for our models to actually do just a little bit on a variation of the light reactions that we need to focus on, and then I think we'll complete the drawing for the light independent reactions. They're not as in-depth, at least the way we cover them, as what we did before. So let me just grab my remote. So quick rehash of where we were at. I'm just going to use the one from the notes. So on Friday, we drew out the light reactions of photosynthesis, which are happening in the thylakoid. And they would be all over the place in the thylakoid, but we're just isolating one system here. And really what's happening is in light reactions, we're doing the first steps to convert light energy into chemical energy that'll eventually end up in the bonds of glucose. So how do we do that? It's kind of like two things going on. One, we got to make ATP. So how do you turn sunlight energy into ATP? Photophosphorylation. Take electrons, energize them, send them down the electron transport chain. That'll keep the proton gradient going, which keeps the ATP coming. So that's some sunlight energy captured in ATP. Re-energize with photosystem 1 because it's in the order it's named, not the order that the electrons flow. And then capture those electrons on a carrier, send that energy to the light independent reactions, aka the Calvin cycle. So that's the skinny on what we had done. And what really is happening here is that light energy, by exciting the electrons and capturing them, is being converted into chemical energy. But NADPH and ATP are not very stable molecules, and you can't really build macromolecules out of them like you can build out of the products of light independent reactions. So we have the chemical energy made. It's just a matter of getting it into a more stable form at this point, aka glucose and other sugars and stuff in macromolecules. So this was kind of like phase 1 of photosynthesis. But that sunlight energy has been converted. Water provides the electrons and the hydrogens that are needed, and the oxygen's released because it's not needed in this process. So that was pretty much the other day. Are there questions? Okay, so one thing about this that we have to draw out, and what you can do is we have like 2 things to cover, is I would kind of, what I would do, because there's not as much to draw for this, we can kind of use the paper a little better. If you take your paper and you just kind of draw a line down the middle, we'll draw like part of this over here, and then you can do the finishing part over here. I think that will work to separate the last 2 things so that you don't need extra paper. If you do need extra paper, I have it, but all right. So on the left side, we're going to focus on the light reactions continued, kind of. And we're going to look at linear versus cyclic electron flow. So there's some change ups that can happen during the light reactions to vary the products that you get as far as the amount of ATP or NADPH is concerned. So we just have to account for those, and that's what we're going to do. So in that half, you can actually split the half in half and just draw a small version of what we had before. So small version, we're not going to put a lot in here. So you can just 2 electron transport chains, photosystem 2, photosystem 1, and thylakoid membrane. Same here. And we'll show the difference so that we can be clear. So photosystem 2, photosystem 1, got our cytochromes in between, and we'll do one side will be linear flow, and one side will show cyclic. So this is like a half of a half. If you mess up, no big deal. We have extra paper. But okay. So when it comes to the flow of electrons, there's some variations that can happen. So like in the case of linear flow, we've kind of already studied this. So water is in the thylakoid space. It releases electrons, which get picked up by the photosystem, and those electrons jump in energy level because of the sunlight. They go down one electron transport chain, and they lose that energy, and then they go get re-energized, and eventually go to an electron carrier. So we've kind of got two things going on. We've got this first electron transport chain, which, like we saw in the daily question, is responsible for keeping the ATP coming. And then we have this second electron transport chain, bless you, which passes electrons to NADPH. So that's their kind of purposes. So that's really what linear electron flow is. You're going from kind of point A to point B with your electrons as they go down the chain and through the photosystems. So if we were to define this, and let me just make sure I'm consistent. We'll put a little note at the bottom. So linear electron flow is the primary pathway, which we just saw. That's why we wrote it first. That electrons follow. It uses both photosystems. So key is it uses both and produces equal amounts of ATP. P is equal, and NADPH. So it produces equal amounts of both of those products. And that's what we saw when we drew it out. We showed linear flow in the drawing that we did on Friday. Now, cyclic flow is a little bit different. What happens here is the electrons, when they get to photosystem 1, they kind of change up a little bit. They go into photosystem 1, they get energized, but then rather than going to be put into NADPH, they go back through the first electron transport chain. Photosystem 2 is not involved, and the second electron transport chain is not involved. So the electrons just keep cycling through photosystem 1 over and over again, getting re-energized and going back through that electron transport chain. Why are they doing that? Well, if you only keep this electron transport chain active, you're only going to make ATP. Here is making NADPH, but that's not going to happen because the electrons never get to there in cyclic flow. So cyclic flow is like a cycle, right? It only uses, photosystem 1 and it produces, or justs, a surplus of ATP. So it's producing extra ATP That's its goal, is to produce a surplus of ATP. Why is that? It's for the Calvin cycle. The Calvin cycle needs more ATP, aka the light independent reactions, than it does NADPH, so it gets the extra it needs that way. So those are just little variations that can occur to get the right amount of products to make sugars efficiently. I'm going to go out on a limb and guess. All right, so that's linear versus cyclic flow. You may see some references to this. We'll do some practice problems that have it in it. And it kind of makes sense, right? Linear is point A to B. Cyclic, the electrons are getting passed back and using that transport chain, to keep the hydrogens going in, making the gradient, making ATP. Photophosphorylation all day long. Questions, concerns? Okay, so that's one little caveat, but other than that, that's all we need to know, in case it's referenced. Okay, so we're at the point now where we're kind of ready to zip this up as far as how the sugar gets made. We saw how the light energy gets converted into ATP and NADPH. Now we just got to kind of get to the final steps. And the good news is the way we cover this, it's really not as complicated, I think, as the light reactions. It's more of a matter of just what goes into these pathways and what comes out. We don't have to know all the enzymes or anything crazy like that. We just have to know one enzyme that's pretty important, and that's it. So on the other half there, or wherever you have space, this won't be as an elaborate of a drawing. It should be a little easier. We've got our final step, which is the light, you can label it independent reactions of photosynthesis. So we can get those out of the way. And then tomorrow, we don't have lab tomorrow, do we? But whenever we don't have lab, we'll spend some time kind of digesting this, because you got to take a minute. So in the corner, you can draw our old friend, the chloroplast. And instead of focusing on the thylakoids this time, we're going to be out here in the stroma. And we're going to zoom in right on that cycle. Extra paper? Just had some there. You can use staple or tape right now. Plenty. So we're going to zoom in on this, and we're going to see the Calvin cycle, which is the light independent reactions. If you want to put Calvin cycle here, just so you know, it's referring to the same thing. Okay. So, all we need to draw for this is a cycle-like model. Nothing crazy. And then we just have to mark out each stage of this. So the light independent reactions don't need light directly, but they do have a couple of steps that kind of make everything happen. So we want to show those. Let me make sure I'm right in the way I put this. I will draw it differently. All right, so we're going to kind of draw sort of a pie chart looking thing. That'll do. So 3 phases to the light independent reactions. And we'll just write them in now. There's fixation, reduction, regeneration. So basically what we want to do here is take the ATP and the NADPH and take the sunlight energy that they've captured and make it into something that can make glucose. So how do we do that? Well, we'll start here and kind of work our way around. We'll call this the beginning, I guess. We're going to have to talk about three chemicals that take the CO2 and eventually get the CO2 to a point where it is going to be reduced to something like glucose. There's a lot of steps and enzymes in this. We're going to skip over most of them. We just have to look at what goes in and out. So one of the first molecules we're talking about is called RUBP, which stands for like ribulose bisphosphate. It's a big long name, so we just call it RUBP. And RUBP has 5 carbons, and we're going to need, I think, 3 of them. Yeah. All right. So RUBP is what we call the CO2 acceptor. And you might have an idea of what it's about to do. So what plants need to do is they need to capture CO2 from the atmosphere and put it into organic molecules. Just like we saw in the carbon cycle, we know they do this. Here's how they do it. So from the atmosphere, there's going to be carbon dioxide that is picked up by the RUBP by reacting them together. Through a series of reactions, they're going to convert to another product, but there's an enzyme that's involved in this, and it has kind of a weird name, but it sounds a little bit like its substrate, Rubisco. RUBP. It's called Rubisco. So the first step here is number 1. Is to capture CO2. We call that fixing it, or basically attaching it. So we can put a little note to ourselves. There's about 5 or 6 steps that we'll break this into. So CO2 is fixed or attached, hence the name fixation, to RUBP by the enzyme Rubisco. So Rubisco is an important enzyme because when it comes to evolution, the evolution of this enzyme changes the game a bit. Because when we talk about producers making carbon available to all themselves and all other living things through the food chain, this is the enzyme that's doing that. So every bit of carbon in our body right now, pretty much at some point, was fixed by something doing photosynthesis into a macromolecule that we eventually consumed or our food consumed, and then we consumed that. So it's a pretty important enzyme that we focus on a lot because plants don't really do the plant thing or photosynthesis thing without it. So Rubisco is the enzyme that's going to get this done. We can put a little note, Rubisco. And now what happens is through a series of reactions throughout fixation, you create a new molecule called PGA, which also has a really long name, so we'll just call it PGA. And you're going to get 6 of these PGAs. And they each have 3 carbons. Now, little side note, if you ever go into more depth on this, just we'll be honest about it. It enters 1 carbon dioxide at a time, but we're going to kind of act like 3 turns of the cycle have happened. So let's make a little note. We'll say 3 carbon dioxide enter. They would do this 1 at a time. So just for accuracy's sake, put a little note in there. One at a time they'll go in. So easy. Step 2, what happens after step 1? PGA is formed. All right, so we've got 6 of these PGA molecules, and if you count the carbons, it kind of makes sense. You have 3 5-carbon molecules here, add 3 more carbons from the 3 CO2s, break them apart, and you end up with 6, 6 3-carbon molecules. All right, now time for reduction. So reduction is exactly what it sounds like. It's time to take all that ATP, take the NAD, pH, and get that energy into the carbon that you want to make products out of, aka CO2. So they're both going to enter in. ADP is going to come out, plus broken off phosphate. NADP plus comes out. So the electrons, the hydrogens, the energy from ATP, all of that's being absorbed by PGA through the reduction pathway, or the reduction part of the pathway. So we can write another step. This one's got a bit more to it, but there's a bit more going on. So PGA, RGA. So PGA is reduced by receiving electrons from the light reactions in the form of NAD, pH, and ATP provides energy. Where'd that energy come from? The sun, right? And the breakdown of water. So sunlight energy in a bottle or in this molecule. So what happens as a result of that? Well, what comes out of that process is a new carbon molecule. You get 6, they have 3 carbons in them. Of these, and it's called G3P. It's called like glyceraldehyde 3-phosphate. It has a long name. We'll just call it G3P. Keep it sane, for us at least. So step 4, G3P is formed, and G3P has all that sunlight goodness energy in its bonds because it was created by the stuff that came from the light reactions. Question? I heard someone say, what's that say? We see it okay? All right, we're almost there. So, made some G3P. Last 2 steps, or step really. 1 G3P for every 6 is removed from the cycle to form glucose. And we can put a little note. And could be lipids, could be proteins, macromolecules, etc. So what does that mean? Well, for every 3 carbon dioxides, basically every 3 turns of the cycle, you make one G3P. The G3P is, I know you might be thinking, wait, that isn't glucose. I thought the formula said glucose. G3P is the building block of glucose. So it can be used to make glucose sugar. It can be used to make lipids. It can even be used to assemble proteins. Remember, plants use this process to make all of their food. You know, including fertilizers they need, obviously, with phosphorus and nitrogen. But the carbon is coming from this process. So they can make a bunch of different things out of it. So G3P is like the raw material that has all the energy that the plant needs. Now, 5 of these G3Ps stay in. So they're still in the cycle, and they're going to be in regeneration. We add a little bit of ATP in to build, which becomes ADP. And where do we end up? Back at RUBP. So if you want to add it in step 6, 5G3P are used to regenerate, hence the name, RUBP, to start over again. So it starts where it ends. That's where it's why it's a cycle. So those 5 that we kept of after 3 turns of the cycle are actually going to regenerate RUBP to keep the whole thing going. And that's pretty much the reactions of the Calvin cycle as far as we need to go into them. One thing I like to note, even though it's not like totally accurate, is I like to say that this extra little ATP at the end is from cyclic flow. Obviously, the ATP is from both processes. You don't really know where it's being put in, but I like to put a little note like, yeah, that's cyclic flow. That's that last little bit of ATP. That's why there's cyclic electron flow, because we need a little more ATP than we do NADPH for the Calvin cycle. And then it'll begin again. So really, to make a glucose molecule, you need 6 turns of this cycle, or 2 with 3 CO2, the way we put it, and that will give you enough carbon to make a glucose molecule. So the direct product are these G3Ps, but they can make glucose if you have 2 of them, 3 carbons each. That's enough to make glucose, which has 6 carbons. So the carbon accounting, you don't have to do too much, but just the basics of what each phase is taking in and producing. All right. So that's pretty much it for us when it comes to the bulk of photosynthesis. We got a little bit of comparing to do back to respiration. We'll kind of spend tomorrow just sort of going back through, making sure like light reactions into the Calvin cycle we get. If you want to take a note, you don't really have to because we put it in the overview. Remember, the ADP and the NADP plus, they're going to all go back to the light reactions, right? So don't forget those products, ADP, NADP plus, they're going to go and do the whole thing again. Just keep picking up electrons from water, getting them energized by light energy, pumping out glucose. So this is why plants need water, sunlight, and carbon dioxide. They're doing chemistry to produce their food and the food that all of us eat too, one way or another. And basically, plants are the thing. Any biomolecules that we have with carbon in them, it was a plant somewhere or something like a plant, like an algae, whatever it is, that captured that carbon from the atmosphere, brought it into this process, and through cellular respiration, what are we doing? We're exhaling that same carbon, and it might be brought in by a plant again. It's just the carbon cycle. So now we can see close up what's going on inside of these organisms. So we'll stop here. We'll rehash this. There is the homework that you can definitely almost or pretty much get done now, which is a good kind of check to see if this is making sense. So if you want to take a picture of your poster, so you can use that later. That's not a bad idea. If you want to grab your phone, you can do that. And then we'll just put the posters back on the drawer. We'll continue on tomorrow.